And I'm excited to introduce our first keynote speaker, Dr. Valerio Hoyos-Villegas. Dr. Hoyos-Villegas is an assistant professor of plant breeding and genetics at McGill University, where he holds a research, teaching, and service appointment. He obtained his master's in crop physiology from the University of Missouri-Columbia and his PhD from Michigan State University in plant breeding, genetics, and biotechnology. Valerio is an associate and technical editor in multiple scientific journals, including the Plant Phenome Journal, and he serves in leadership roles at the National Association of Plant Breeders and the North American Plant Phenotyping Network. And I know personally that Valerio has really gone above and beyond to put Canada into the North American Plant Phenotyping Network, so we appreciate his efforts on that. Today's presentation will focus on the work that his pulse breeding and genetics lab at McGill carries out in the advancement of phenomics and genomics for the acceleration of genetic gain for the development of pulse crop varieties. So please join me in welcoming Dr. Hoyos-Villegas.
Thank you very much, Professor Stavnes, for the kind words and introduction. I'm very happy, very pleased to be here to get the opportunity to share some thoughts with you. So the title of my talk is Turning the Phenotyping Bottleneck into a Pipeline. So in plant breeding, we always talk about high throughput phenotyping bottlenecks and how do we alleviate these bottlenecks. But we also talk a lot about breeding pipelines. So this is a bit of a play on words on how these two conditions intersect with high throughput phenotyping and plant breeding. So an overview of my talk, I'll start with a bit of introduction about the background and the challenges that we face in bringing forward approaches such as digital agriculture, phenomics, in the context of plant breeding. I'll talk a little bit about the role or some thinking I've done about of phenomics and plant breeding, as well as a bit of some research that I think is exciting from my lab. It's not primary domain phenomics, but I think it's worth highlighting, and also some general conclusions. So historical increases in yield. First, we'll do a little bit of background around this, if you'll allow me. Many of you are familiar with this formula, but if you are not, this is generally regarded as the breeder's equation on the response of selection equation, or the response to selection equation. Response to selection equation is made up of a few terms that, when maximized, result in a higher rate of change due to selection. These terms involve selection intensity, selection accuracy, additive genetic variance, and years per cycle. Our ability to introduce logistical and technical efficiencies to the way these parameters operate in the overall structure of a breeding program enable us to deliver higher response to selection. These can range from larger populations, higher noise-to-signal ratio, to enhanced allelic diversity, or reducing the number of years per breeding cycle. Here we have some examples that I compiled from a few sources showing the success of breeding programs on increasing yield. As you can see, many of these examples show an increase of 1 to 3%. Plant breeding has enabled significant gains in yield and other characteristics for multiple crop species. Plant breeders and crop sciences, we face the challenge of increasing food supply in order to meet a growing population. We can address this by aiming to close the gap that exists between on-farm yield and experimental or potential yield. From a breeding perspective, increasing the rate of genetic gain will translate in the closure of the yield gap. So the big question for plant breeders is, how do we further increase rates of yield gain? In order to understand how to increase yield gain, I think we must first understand where yield gains come from. Although practical, it should be noted that regression-based estimates of yield gains are not in proportionality to response to selection. This is because of another well-known framework, which is that phenotype is equal to genotype plus environment by genotype by environment plus environment. The environmental terms are therefore non-genetic terms and do not contribute to sources of additive increases from genetic effects. Therefore, heritable increases in genotypic value are the sole source of genetic gain. As a bit of review, we can consider the concept of average effect. If we consider the population with gametes uniting at random, the mean set of individuals receiving the same allele and the mean of the population of gametes united at random, including the allele of interest, will determine the average effect of an allele. In practical terms, if we subtract the progeny mean from the population mean to determine the relative breeding value contribution on the set of parents that make up a breeding program. In a plant breeding context, practically speaking, increasing the average effect of parental genes is associated with genotypic value of the progeny, which will become the next generation of superior parents. Thinking in this framework, we can surmise that increasing the frequency of high breeding value candidates in a plant breeding program is only possible with accurate estimations of genotypic value. Measuring genotypic value from additive genetic variants can be approximated by utilizing linear models. A linear model for environment I, for phenotype I in environment J, for example yield, will equal to a grand mean plus an environment, genotype I and environment J plus genotype I and environment J plus an error term. Therefore, in the context of using phenotype as a response variable, we can, 1, aim to utilize an estimation of the genotypic value to improve our estimates of or predictions of phenotypic value. However, biology affects our assumptions when predicting phenotypic values. That is, under complex trait scenarios, key assumptions do not hold, such as large numbers of QTL, no major QTL, and evenly distributed QTL. The second approach is that we can try and minimize the environmental effects and their interaction as close or equal to 0. On both cases, accurate phenotyping is highly important. On one end, it enhances SNP-based signals and predictions, and on the other hand, it minimizes environmental noise. So what is the role of phenomics in understanding complex traits? To answer this question, I thought it would be worthwhile reviewing where are we in applications from genome-wide association for plant breeding. Genome-wide association has enabled us to develop gene-based markers, such as SNPs and indels. Genome-wide association datasets have become easily shareable and consolidatable. And publicly available data facilitates these novel discoveries. Also, genome-wide association has enabled us the estimation of genetic correlations and interactions between loci at the genomic level, due in part also to this shared datasets. Finally, genome-wide association has enabled us to obtain estimates of SNP heritability for complex traits. However, many challenges still remain. For example, my student Henry Cordova extracted a table from this review, genome-wide association studies in maize, praise, and stargaze, of historical reports of GWAS across about 15 years, and added a column with the reported number of hits for each one of these studies. A few conclusions can be drawn from here. One, locus rates continue to increase, and this is in line with the fact that for reference, human loci detection has yet to plateau. Across approximately 80 studies, we can also see that increases in 2 to 3x in panel size have a much greater effect than increases in 10 to 20x in marker density. So diminishing returns on marker set size are not the complete picture. Genetic architecture and low frequency variants still limit detection power. But increasing panel size do enable further discrimination among different levels of genetic architecture complexity. These last 2 points emphasize the need to improve our phenotyping throughput in order to achieve detection in practical terms. For example, I feel it is important to highlight the recent publication of missing heritability for human height. Essentially, the full extent of known human heritability has been covered. This is in the backdrop of the human genome, full genome sequence release, where upon release of the human genome, height heritability was about 5%, leading to the question of missing heritability coverage. Now, approximately all of the approximately 50%, which is the proportion of variance due to genetic sources of height heritability, has been accounted for. However, this required 5.54 million individuals sampled in a combination of 281 genome-wide association studies, and this is mostly only applicable or variants from European populations. This highlights the need for larger, higher resolution collaborative research projects in phenomics and genomics in plant breeding applications. Moving on, I think that in order for us to better gain knowledge and extract suitable applications from phenomics, we should also consider the dynamics that genotype and phenotype spaces carry out under natural or artificial breeding populations. On the right, we can see the two planes, the two dimensions, where a genotype J placed along a continuum of potential genotypic variants has a corresponding internal phenotype, which is modulated by external environmental cues or influences. These external environmental conditions determine a particular phenotype to gain or lose fitness and functionality, creating a new phenotype, for example, P'. By consequence, this P' phenotype will be linked to a different set of genotypic effects, or G', moving back up to the genotype space. Through artificial or natural selection, this new set of genotypic effects will experience a shift, either positive or negative, in frequency through systematic or dispersive processes, for example, mutation, selection, drift, etc., leading to a new genotype, G2, and so on. Therefore, due to levels of organization and nonlinearity between these two different dimensions, any suitable model must be able to incorporate functional covariates constructed among multidimensional genotype and phenotype spaces. Some examples of models that enable high-dimensional data could be implemented in the future are structural models, partial least squares regression, random forests, support vector machines, and functional regressions. Many of these models share advantages and disadvantages, but some of the disadvantages involve loss of features or dimensionality, prohibitively large sample sizes, and estimated reliability and convergence, or are simply computationally intensive to resolve, particularly given the data volumes that we deal with in phenomics platform. So how do we deal with these challenges, and what is their role in determining applications for plant breeding? Just as with genome-wide association, phenome-wide maps can relate to relative distances and relationships that exist among traits measured using high-resolution sensor approaches. This brings me to discuss the potential that exists for merging genome-wide association and phenome-wide maps. Given the high nonlinearity of phenotypes and genotypes, there is relatively less value in utilizing individual genotype-phenotype associations in order to advance selection or prediction for a complex trait. It is unknown whether the incorporation of phenome-wide maps will enhance prediction, yet it is still more useful for breeders to think in terms of total additive or genetic variation and how this can be comprised as a summary of effects of multiple intervening phenotypes for the final trait of interest. For example, utilizing genome-wide association peaks to inform genomic prediction does not result in major improvements when compared to changes in the optimization of training and test sets. Therefore, the intersection in functionality among genome-wide association and phenome-wide maps datasets may prove particularly useful with better elucidation of pleiotropy. Pleiotropy happens when there are individual genetic loci that harbor variants that are associated with multiple distinct traits. That is, one locus equals multiple traits. Similarly, cross-phenotype associations are genetic signals in linkage disequilibrium co-localizing to loci responsible for multiple traits. It is important to note that pleiotropy cannot fully be explained by cross-phenotype associations, but it can be one cause. For example, some of the examples are below. Looking forward, novel phenome-wide maps will enable higher accuracy in estimation of cross-phenotype effects. One such application of this improvement is on genomic predictions models accounting for multiple phenotypic outcomes. As a model, ideally, we could utilize a similar approach to high-throughput-based selection as we would with marker-assisted selection. That is, if we consider a routine QTL mapping procedure where we have a large QTL that are responsible for a certain final effect of the trait of interest, we can imagine scenarios where phenomics and phenome-wide maps can contribute. First, we can think of multiple intervening QTLs of variable effects which will be focused solely on traits that are in association with the trait in aggregate. Second, phenomics might enable us to increase the power to detect small effect QTL that are related to the aggregate trait, in the same way that QTL may have positive or negative activity or effect due to coupling or repulsion phase linkages. These dissected QTL can have difference in magnitude due to directional pleiotropy, but this can be accounted for utilizing statistical genomics approaches that we can use to remove phenotypic complexity and identify cross-phenotype associations. Therefore, in order to make gains in complex traits, it is possible that we can rethink of ways of conducting selection. Phenomics will enable us to account for a greater proportion of heritability by using dissected traits, then utilize them in aggregate to rebuild a phenomics-based trait of interest. An emergent area that has been developed partly to address large P, small n problems in phenomics have been latent space phenotypes. Latent space phenotypes, as many of you know, allow for measuring traits outside the conventional frameworks, for example, geometry, through the identification of appropriate decoders and encoders. LSPs enable identifying new traits across multiple dimensions by reconstructing on the basis of multiple latent dimensions. Some thoughts on LSPs. One obvious challenge for the implementation of latent space phenotypes will be drawing conclusions on biologically relevant information. That is, principal component analysis, for example, may allow us to reconstruct phenotypes, but they still need to be relevant in a biological context, which is challenging when we are dealing with multidimensional data. However, LSPs may enable us to discover new traits through examination of integration of features suggested by trait-agnostic models. The following question is regarding loss of information. We have highly dimensional data, but with relatively small sample sizes, particularly around traits that are not easily distinguishable by sensors. So it will be challenging to perform trait aggregation as well as to retain features and models that utilize dimension reduction approaches in the bigger picture. One such possibility is that ridge regression models, for example, can fit models without dimension reduction or transformation. Therefore, latent space phenotypes may be less user biased and provide us with a means of complementing datasets among relatively obvious plant characteristics. However, how will we approach the problem of integrating traits that involve intersection of biochemical, physiological, morphological, and environmental spaces, such as yield and other agronomic traits? An early form of LSPs as integrators of traits has been done through the incorporation of a Weibull proportional hazards model in animal breeding. The use of proportional hazards model included multiple traits as part of a likelihood function to estimate survival. The results then showed that at higher final scores estimated from the model would result in lower risk of culling. In order to begin incorporating these types of approaches in the field of crop breeding, we have constructed the McGill Field Phenomics platform as part of the ECP3 project, which is the Eastern Canadian Plant Phenotyping Platform. Our platform is mounted on a high-clearance sprayer, which enables us to accommodate multiple types of crops. The system is mounted in front and back of the vehicle, allowing us to measure morphometric imaging, light detection and LiDAR, visible light analysis, chlorophyll fluorescence and kinetics imaging, thermal, hyperspectral, multispectral imaging, as well as RTK navigation and in-canopy weather sensors to account for spatial variability. For example, an early incorporation of phenomics in my breeding program will enable the use of canopy reconstruction at the single plant level in early generations and under a row crop configuration at advanced generations, in addition to other to genomics platforms. An FRQ&T funded project, which is the Impulse project, will enable the advancement of these types of efforts. It involves the development of new field phenomics algorithms for the detection of stress tolerance in segregating populations, and also the development of canopy reconstruction and yield estimation phenomics tools. It also relates to the construction of collections and information to develop the basis for breeding for durable white mold resistance or tolerance or physiological tolerance. The objectives 1 and 2 then link with objective 3 in the construction of regulatory networks, and it also will conduct an economic analysis of the expansion of pulse crops in Quebec and Eastern Canada. Switching gears a bit, some phenomics related research from my lab. One such effort has been the investigation on water use patterns at the whole plant level in common beans over long-term periods. My student Henry investigated the genetic architecture of whole plant transpiration and water use in common bean by analyzing water deficit thresholds, inducing stomatal closure. This project also intended to develop an inexpensive and semi-automated system to measure compounded rates of water use at the whole plant level. In this study, we utilized a relatively small association panel, but a high-frequency data collection system. The method we constructed was a simple, inexpensive, automated system that would enable us to estimate daily water use for drought and control treatments, as well as calculate replenishment amounts from transpiration in the control. Without such a system, estimating daily plant water use across a long experimental period would not be feasible. By collecting water use in relation to total of extractable soil water, that's the FTSW estimator, we are able to obtain a normalized transpiration rate in relation to an irrigated control. Such a system enables us to approximate to the slow rate of development that drought has under field conditions and the plant's response to this process. The behavior of these two variables enable us to fit a nonlinear model to the data and estimate a point of stomatal closure. On the right, some of the results. On the right, we can see the behavior of the top and bottom genotypes displaying early and late onset of stomatal closure at different levels of FTSW-C, which is the threshold at which stomatal closure begins to happen. On the bottom left, we can see the individual critical values of this inflection point after fitting a nonlinear regression. By taking all the individual inflection points of FTSW-C and rates of transpiration, we conducted genome-wide association on them, and we found a number of promising signals, including transcription factors and aquaporins. The signals and genetic variability found in this study are promising leads for the development of drought tolerance in common beans, but functional validation is of course needed. So simple and inexpensive methods of measuring whole plant physiological traits enable collection data at larger scales of genetic variation. The last project I will talk about is an area I refer to as endophenomics. In this example of an endophenotype is the use of bendable sensors for phenotyping pH-related responses in common bean. This is the work of my former student Jennifer Lin with our collaborator Professor Dahia at the University of Glasgow. As many of you may know, the invading fungal spores of the fungal pathogen causing white mold Sclerotinia sclerotirum germinate on decayed tissue and produce hyphae that penetrate the surfaces of the leaf. The fungus will then produce organic acids in the form of oxalic acid or citric acid, and this in turn lowers the extracellular pH and surroundings, creating an optimal environment for cell wall degrading enzymes that secretes and enabling further infection. So bendable pH sensors have previously been used and validated for biomedical purposes. They measure voltage of a solution or a substrate, and voltage in these sensors has been calibrated with pH of certain surfaces, such as human skin. The voltage related to pH is therefore the potency of charged hydrogen ions in solution, so they can be easily calibrated to a particular range in voltage. We wanted to know if these bendable sensors had applications in plant tissues. Among all of the various methods available for measuring pH, we used apoplastic fluid for validation. The objective was to explore if there are differences in disease score, apoplastic pH, and voltage among leaves in of infected common bean varieties with different levels of white mold susceptibility. A standardized system for estimating disease score was determined using the straw test method, which involves cutting the main stem and placing inoculant over it to produce infection. We then assess the plants 8 days later in a score given from 1 to 9 to determine, with 9 being the greatest level of disease. We used 8 varieties with various levels of resistance. To use the sensors, we utilized a simple Arduino platform to take voltage readings, and then the sensors were attached to leaves at the infection site. Readings were then recorded at 60-second intervals. First, we tested some of the different sources, different types of reference electrode materials provided to us by our collaborators at the University of Glasgow. We found that the ones with carbon as a reference electrode had the highest sensitivity. However, some hysteresis or memory effect was found. Some results from bendable sensors where we measured voltage. So the orange and blue line indicate voltage changes that were consistent across mock-inoculated and inoculated treatments. So time of treatment was definitely statistically significant, and both variety and susceptibility groups were statistically different. Among mock-inoculated and inoculated treatments, 5 out of 8 cultivars showed consistent pH and voltage readings. Straw test scores confirm findings from the literature. Apoplastic pH following infections increased in all of the susceptible ones. But a minor correlation was found between voltage and the straw test score, so this is still as work in progress. However, this small amount of success and validation in plants has led us to securing funding for the Tesla project, which is Transient Electronics for Sustainable ICT in Digital Agriculture. And yes, that is an acronym inside an acronym. This has led us to success, which is a multi-partner project aiming at developing bendable sensors for digital agriculture. The ultimate goal is for the sensors to be disposable, self-powered, and biodegradable. So in terms of some general conclusions, breeding value-oriented phenotyping will be the most important characteristic of the implementation of phenomics in order to make genetic gain. When we harken back to the early days of phonetics or the concept of a phen related to as the basic unit, we can note, or I have noted, that a genome-wide assembly is tractable in the form of individual genes, but a phenome is not. So for us as a community, my question is, what is the basic unit or building block of a phen? In the medium term, I think that the influence of the role of phenomics may likely be in the plant breeding may likely be in the pre-breeding space or the allele mining space. For example, highly contrasting phenotypes. But attempting to take a phenome approach may lead us down the road of detecting common variants, such as the case of early applications of genome-wide association. Bioinformatics pipelines and models enabling multidimensional data analysis are now available, but we still have the data volumes bottleneck. AI and machine learning will play a key role in extracting functionality, for example, LSPs and phenomics. But we don't know once we incorporate these systems into plant breeding programs, whether that will cause loss of favorable alleles. That will have to be tested and validated. But the challenge remains how to construct appropriate phenome-wide maps. So final question, can biological outcomes be most accurately predicted by the phenotype? Right, i.e., the phenotype the phens determining the phenotype and their fitness, and not individual genes. Finally, I'd like to leave you with this thought. On the left, we have the initial phenotypic map that was developed by R. Prakken at Wageningen University. This phenotypic map was developed through meticulous crossing and evaluation of seed code colors and color variations in order to establish respective loci and loci modifiers. It was a phenotypic map. Seed code colors in common being have very high heritabilities and serve as a good model to study linkage and interactions among loci. Fast forward 77 years, we now have a common bean reference genome which encompasses coding and non-coding regions, many with putative or validated functions. This has been a tremendous gain in increasing bandwidth between the genotype and the phenotype. However, if we compare the number of seed code color genes cloned and or sequenced before and after the release of the full reference genome, these only account for less than 20% of the variants that Prakken originally reported in his many studies. Indeed, our understanding of the phenotype-genotype relationships remain largely an unexplored world. Acknowledgements to my lab, staff, grad students, my funders, because they are really the backbone of what I come here to talk to you about, and many thanks.